So I'm pleased to introduce Dr. Guillaume Butler-Laporte, a clinical scientist based at McGill University Health Center. Dr. Butler-Laporte began his academic journey at McGill, where he studied mathematics and statistics before pursuing medicine at the University's medical school. After completing his residency in infectious diseases and medical microbiology, he then dedicated himself to research, contributing significantly to the field during his time at Research Lab within McGill's clinician genomics program from 2020 to 2022. Dr. Butler-Laporte was then a CIHR research fellow at University of Oxford, further expanding his knowledge and networks before starting as a researcher at Lady David's Institute in this January. As an alumni of McGill, Dr. Butler-Laporte brings a wealth of expertise and dedication to his work, making him a valuable asset to both universities and the broader medical community. Please join me in welcoming Dr. Guillaume Butler-Laporte.
I need to approve that it's recorded. Hold on. Well, thank you, Alina. Thank you, everyone. Good. And you can call me Guillaume too. It's fine. All right. So I'm very happy to be here because as Alina said, I just started literally here and then was recruited, or actually I approached Celia to be recruited, to be fair, to the QLS program. Yeah, roughly in January. So I thought it was a good way for you guys to know about me. This is actually, I wasn't entirely sure, and that's one of the things I want to note. I'm going to try to see today. I wasn't sure like how everyone was comfortable with genetics. I know it's a quantitative life science program, so it's a very broad field. So I actually took the approach of not expecting you guys to know much about genetics, and I'm going to try to make this as non-technical as possible. And one of the reasons is that I presented something similar about the HLA to the LDI clinical epidemiology group, and I think I completely lost them because I hadn't realized they knew nothing about genetics. So I'm taking a completely different approach with you guys, and I'm going to expect not much, and try actually to make it very little in terms of technical knowledge and expertise, and really just try to show you what human genetics is doing and how it translates to clinical medicine, and try to actually be your cheerleader, guys, during this PhD, and try to make you see that actually what you guys do can have great impacts on what we do clinically. So if it can actually encourage you to continue when you're wondering about your career, well then great, that's one of the goals today. All right, so to answer that question, yes, can genomics really improve management of human disease? Yes, that's the goal. I'm going to hope to convince you of that. The reason why we're going to review what we did during the pandemic is because the pandemic was a great natural experiment in that for all researchers, the clock started at zero. And we were all doing our things in different silos, and so that was a great opportunity to essentially honestly compare what was our results in terms of translating to actual clinical practice. So while the clinicians were doing their trials, sometimes a bit at random, they were obtaining certain results, and we were doing the same in genetics, and so you could actually compare in real life without cheating. And see if they actually, well, one predicted the other, because that's what we want to do in genetics. So we'll see how we performed. So, all right, this is the game plan for today. Just a little recap to see where we actually stand as a role as geneticists. You know, in genetics, the first step is usually to try to target the disease. In this case, is an intubated patient with COVID-19, try to find the genetic determinants of it. Then we try to turn them into pills. And then we'll let usually the clinical trialist turn that pill into a trial to see if it actually works. So our goal as geneticists is, I don't know if you can see the arrow. Can you see the arrow? Not at all. Okay. The first two arrows here and there, try to optimize them so that the third arrow back to the patient has a higher chance of success. Easier said than done, of course, but that is where in the QLS program, all of us, that's where we actually try to work. And so we'll look at these two arrows specifically and then compare to the third one at the end. All right, disease to variants. So, yeah, many of you seen a graph similar to this. I mention it because there's still, when people think about genetics, and especially clinician, but a lot of people who did more biology, pure biology undergrads or microbiology and things like that, they still think a lot about genetics, about like looking at rare genetic variants affecting very strong extreme phenotypes called Mendelian disease. So one genetic variant leading to one bad disease. But really it's more of a spectrum, right? Every one of us carries variants that other people carry also, and they all have different impact on the phenotypes in question. So COVID-19, for example, but they each carry kind of their own problems if you want to analyze them. So if you're looking at rare variants, so again, I can't, sorry about that, but if you look at,
You should be able to. Click on the.
there you go. There you go. So if you look at rare alleles causing Mendelian diseases, because those alleles have a very strong effect size and cause an extreme phenotype, you'd expect not to find a whole lot of them. Anyway, if you look at the other end, the common variants, you find them in a whole lot of people, but of course their effect on any phenotype is modest to low. And here, if you actually follow and backtrack the common disease, common variant bubble all the way to the y-axis, it shows intermediate, but really in practice, it's really low effect to modest effect size that we see. But there's a whole spectrum there. And I want to emphasize this because in QLS and in the quantitative field, our expertise is actually probably better used in the common variant field. But still, you're probably aware, depending on your background, about the rare alleles causing Mendelian disease, and they do have a role to play, but we're not going to talk too much about it today. Just to say that one of the reasons it's maybe a bit less interesting for you when we're going to focus on common variants is the phenotypes sometimes are a bit extreme and difficult to actually translate to clinical practice. So this is one specific example of an inborn error of immunity, chronic mucocutaneous candidiasis, which is caused by a very specific loss of function or at least a specific class of loss of function at the STAT1 gene, which is usually carried in families. So in the middle, you see family pedigrees, and it causes invasive disease caused by a specific, very common germ called Candida. It's a fungus. Causes skin infection and gastrointestinal tract infection, and this should not happen. Like everyone carries Candida on you, and so this should not happen, and it's caused by STAT1 problem. And one of the problems, of course, is well, this is an extreme phenotype. How much is this really applicable to regular patients with candidiasis? As I said, it is a common condition. More than likely, most of you guys will have some sort of Candida infection in your life. Will probably not be severe. And if we're going to treat you like we treat the chronic mucocutaneous candidiasis, you're not going to like us. So you know, yes, inborn errors of immunity or rare variants in Mendelian disease can inform a lot about biological pathways and translate clinically. And there has been clinical successes like this. On a larger scale, it's a bit difficult to actually think of whether this applies, well, if this applies on a larger scale is what I'm trying to say. And of course, for you guys during a PhD, if you're basing it on finding rare variants, essentially it's just luck, right? Are you going to find a family of people that carry a highly deleterious variants in 4 years in QLS program? Unlikely, right? So I'm trying to convince you also that probably not the best way forward for you guys. Not that it can't be done, but you need to be lucky. All right, so common variants. On the other end of the spectrum, can we use those common variants and, in this case, large biobanks, because we need statistical power to find new treatments? I'm going to try to convince you that that's what we did during the COVID-19 pandemic. Yeah, the problem here, just to come back, is that, as I said, because the effect size are small, So low to modest, you need a very big sample size. And big sample size means big data. So for you guys, that's what you want, right? That's where you actually can shine. All right, so this is where I'm going to start to talk about COVID-19, where during the pandemic, very early on, with Brent and with Sylvie also was going to present a bit on that in the next couple of weeks. I tried to coordinate with her so she doesn't repeat what I'm saying. So Sylvie, Tomoko was the QLS program, Tianyuan, a lot of faces you might recognize. We became part of this very big consortium, the COVID-19 Genetic Initiative. Just gonna, can I just?
Yeah, under more, you should be able to minimize
Yeah,
that taskbar.
It's not leaves. Anyway, there you go. I just can't actually move. Okay, fine. So this was a really large consortium. This actually is the map from the first Nature paper. But the second, the third Nature paper that came out of this, the second update, there were like 2 million participants overall. So a very large sample size. We were this little kind of flower there. Okay. But still, you know, like it takes a lot of people, a very large consortium, and we all contributed. But yeah, this was the BQC19, so Quebec's COVID-19 biobank, which towards the end we had around 4,000 participants, so not too bad. Anyway, so we all pooled effort together, enroll as many patients as we can, and then we did. What did we do? Sorry, that can work. There you go. We did what's called a genome-wide association study. This is nowadays probably the first step in common variant genetic association studies. I say this a bit laughing because it's been used and misused, but if you, because it's very easy to perform, but if you do it well, and it can actually really provide nice biological insight into disease, and then do next part later, which is to translate it into medication. So if you know a bit about genome-wide association studies and Manhattan plot, because that's called the Manhattan plot, and I'm going to describe it, you'll notice that this in May 15, we didn't find any signal whatsoever. Okay, forget about the blue line. Essentially, this is a completely null genome-wide association study. So this was in May 15, 2020, so still early in the pandemic. We had only in the consortium about 1,300 participants, with roughly 50-50 cases in control. In those cases, in the next few slides, the cases will always be the same. So people with COVID-19 that are hospitalized. But you'll see that the cases and controls will also increase significantly as we move forward within in the pandemic. Here, genome-wide association studies, so on the x-axis, you have all chromosomes. For some reason, some of the numbers were skipped, but they're all there, except Y, actually. On the y-axis, this is negative log 10 of the p-value, which meaning that, for example, if you have like the points that I'm pointing, like every point is a genetic variant, this is roughly 10 to the -6 p-value, okay? Again, because we're testing millions of variants, like each point is a genetic variant. This doesn't pass multiple testing threshold. Actually, Celia has worked quite a bit on this, I think, a long time ago to determine what was statistically significant in the GWAS. But I can tell you none of this is. But essentially, what we do is at every single one of those variants, we run either a linear or logistic regression.
So Peter, they would have, all these guys would have had a little bit of a module
Very good.
on GWAS.
Just to be, okay, great. So I'll stop there. So this is what happened when we move forward in the pandemic. Okay, so as the number of cases increase, now to 3,000 with controls 900,000, now this red line with this genome-wide significant p-value, you can see it, and you can see that the chromosome 3, there's something happening there, which interestingly, if you were watching a bit the news in the pandemic, that was reported that as that famous Neanderthal haplotype that we inherited when we Homo sapiens mingled with the Neanderthals thousands of years ago. Further then in the pandemic, becomes even more significant and it replicates in every single cohort. Even more, this was the first paper. You'll see here that the y-axis, actually we changed the scale. So it's not just minus log, it's log of minus log. So that's even more squished. And if we were to actually put in the same scale, that chromosome 3 hit would be actually outside the slide. So we continue, even more participants, continue again. This I think was the first update. And this was, I think, yeah, this is from the meta-archive paper, but now it's published, the second update. Yeah, so 40 loci associated with high risk of hospitalization with COVID-19. I know technically there's pros and cons to p-value, but I think the signal is pretty clear here. There's an association that's not only due to pure chance that replicates in multiple cohorts in many areas of the genome. Yes, so what do we actually do with this now? Right. So now we have that first arrow. We went from that sick patient to the variant underlying it. Now we'd like to discover the pills there. The main problem is that GWAS are still an observational study design. Okay, we didn't randomize someone to have a G in position 10 million at chromosome 10. It is purely observational. It can be confounded. You can have reverse causation. There's a whole bunch of different biases that you can think of. And so you can't really use that directly for drug discovery or translation to a specific drug. You need an extra step. And so what's this extra step? Yeah, now that Celia mentioned, maybe you did, we talked already about MR a bit.
I don't know that you guys, of course, some of you have heard of MR, but did you have MR in the course?
Yeah.
Phyllis covered, yeah?
Yeah, Daniel told us.
Daniel, okay.
Okay.
Yeah.
I'm gonna talk about it again. And you know what? I take a different approach actually, because well, first I always like to talk to start with this slide there, because I thought it was funny. It was published as a news feature in Nature. So, one of those, it's not a scientific paper, it's just like a review of the literature, but more like a news story. Like right as the pandemic was developing, December 10, 2019, and now like given what we were about to do with it, that was very interesting. And I like that they called it a gene-based hack, because the first time I heard about it myself, like, this is just mathematical trickery. It can't be right. It doesn't like that. It can't. And yes, you can definitely misuse it, but you can do great things with it too. So the more technical aspect of it, and I think Celia is going to talk about it also with her specific example, but we're just going to scratch the surface and actually compare it with what we did clinically. Just a small review of what it is, of course. This is the way I like to explain MR, which is completely not using genetics. And it's using, because this is like an example that's in all textbooks, not MR textbooks, but instrumental variable analysis textbooks. And it's also something that like, it's been ingrained in us since like primary school that smoking is bad. And so we kind of understand the mechanics behind it, the medicine behind it. It makes so much sense to us. But yet it's not that clear, right? Like if you put yourself back in the shoes of an epidemiologist in the 1950s, and you're thinking, oh, there's an association between smoking and lung cancer, and you're like, is there an actual causal effect? Should I go to the public health authorities and ask them to invest in smoking prevention campaign? You could do that, but technically, this could all be nonsense. Okay, if you want to be very honest, in the 19th, it doesn't make sense today. We know it's wrong. We know it's definitely causal, but that you could make an argument for the opposite, right? You could say that people who are predisposed to lung cancer for whatever lung disease, due to whatever lung disease, they actually look for smoking to clear their lungs and therefore make themselves feel better. And if they weren't smoking, they'd develop more cancers. And this is what the smoking, the tobacco industry was actually saying. And then there's a whole bunch of confounders. Unfortunately, smoking and lung cancers are both associated with lower socioeconomic status, which is probably the #1 determinant of health outcomes worldwide. And so like you can think of a whole bunch of other reasons why this is completely biased. And so what they did, and this was the start of instrumental variable analysis, is they thought, if we could find some sort of a variable that was only associated through lung cancer through its effect on smoking, therefore bypassing confounders and really going in that direction from smoking to lung cancer, maybe we could measure that orange, yellow arrow there. It's more green on my screen, but anyway. So maybe we could do that. And what they thought of using was cigarette taxes in different jurisdictions. So if you go in Ohio, I guess, or whatever, and you see that there's more cigarette taxes, and that's associated with less lung cancer, it doesn't make sense biologically. So it can really only be through its effect on smoking. And if you go to Saskatchewan and there's less taxes and more lung cancer, well, then again, it should be through smoking. So this is actually MR. It's the exact same thing, except our variants, our instruments are genetic variants. Okay, that's the exact, it's the same thing. So that's why I like this example, because it seems less like a genetic hack, less like a mathematical trick, and all of a sudden it kind of makes sense, because it's the exact same thing. We use genetic variants that are associated with, in this case, some sort of proteins in your circulation, and is not associated with COVID-19 through any other pathway than that protein. And then if you just do a bit of math, you can infer the effect and whether there's an effect between the protein and COVID-19. So yeah, so the genetic variants are chosen based on GWAS. So we usually do two GWAS, right? One on proteins or whatever exposure, and the other on severe COVID-19, and then we do MR. So the severe COVID-19 GWAS, we use the one that I showed in the previous section. All right, therapy to patients. So again, this is a nice thing about the COVID-19 pandemic, is that in practice, there's a delay between Mendelian randomization studies and clinical trials. But during COVID-19, well, there were almost none. And the people who were doing MR couldn't, we couldn't go, oh yeah, our method works and replicates what this trial did 10 years ago, therefore we should use MR. We had to commit ourselves to an answer and then wait a few weeks later for the trial to come out. So we had to stay honest. So let's see how we did. All right, so first, some negative controls, right? It's always nice, and because the punchlines, of course, the positive results later. So we'll start just with the T's, the negative controls. So some MR studies, and I chose two because I happen to have published them, but some MR studies that predicted that this specific intervention was not going to work for COVID-19, and the trials said exactly the same. So this one, you probably would not remember it, but it was a big scare among the clinicians. COVID, so the SARS-CoV-2, its spike protein attaches to the ACE2 receptor in the lungs and then invades like this. And ACE2 is, so it's angiotensin converting enzyme 2, which is closely related to ACE1, I guess. And the inhibition of ACE1 is one of the most common ways in practice we treat hypertension. So it is a very broad class of medication. So there was a big scare actually early in the pandemic that people who were on ACE inhibitors could be at high risk of severe COVID. And because it's purely observational, there were other clinicians who said, no, what we should do is people who come in with severe COVID, we need to give them ACE inhibition. So a completely opposite point of view. When we actually show the MR first, when we did the MR, what we predicted was that there was going to be absolutely no effect. And that's exactly what was found after multiple meta-analysis of different ACE inhibition randomized trials. So yeah, nothing at all. Second one, vitamin D. And if you guys want to receive hate mail, I suggest you write a paper that shows vitamin D doesn't work. Yeah, that was funny. Or just go in you guys go on Reddit, the subreddit science, and then just type your name and paper. You'll have fun. The comments sometimes, wow, that was funny. Okay. All right, so for vitamin D, there's two, as far as I know, there's two big trials that were done. One was giving vitamin D to people who already came in very sick with COVID. They gave them a big dose of vitamin D, showed absolutely no difference in outcome. And the other trial was, they took a different approach. They gave vitamin D as a prevention measure. So they essentially enrolled people and waited for them to develop COVID and severe COVID later, didn't do anything. So two trials, two different approaches, vitamin D, didn't show really anything. Our MR, without going into detail, like these are all the different analysis, but like the null line is the dashed line there as one. MR did not support any role for vitamin D in COVID-19. Right, so now the question is, are all MR studies negative? And I chose 3 out of the 6 treatment classes for which we had randomized controlled trial evidence that they work. The other 3, I'll talk about it later. It's not that they're negative, it's more that they're inconclusive, but these 3, we had MR studies and we can actually compare them, okay? So the first one, and I listed them in order of like how much we use them. So IL-6 inhibitors are the ones that are more commonly used. I prescribed it 2 weeks ago. TYK2 inhibitors is usually when IL-6 fails, and interferon lambda was kind of the trial that was done towards the end of the pandemic, so it never really caught on, but it's still, you know, it works. So IL-6 inhibition, which is usually done using a monoclonal antibody called tocilizumab. By the way, guys, if just to give you an idea, all the drugs that finish with mab mean monoclonal antibody, so that should give you an idea. So the idea from the clinician point of view, again, like they weren't basing this on any genetic evidence. They were just going based on basic biology. They were saying, well, you know, SARS-CoV-2 starts a whole inflammatory cascade that seems to converge towards IL-6. And then IL-6 from there, it then diverges again towards a whole bunch of inflammatory cytokines and other inflammatory cascade, and that this overall cascade of inflammation is what's detrimental to COVID-19. So let's block IL-6 and see if inflammation improves and see if the outcomes improve. And that's exactly what happened, actually. Now, what's interesting is in September 25, 2020, so that's way before the trials came out, one of the first MR actually on COVID-19 came out, and they were saying exactly this is if you they were using CRP as a proxy for IL-6 because IL-6 is a bit difficult to measure biochemically, but essentially they were saying if you decrease CRP, which is equivalent to decreasing the activity of IL-6, you improve mortality and hospitalization. So that was like what the lockdown was in March, April, so that's 6 months later, and then a few months later the RCT came out and said exactly the same thing. Second one is baricitinib, which classically in the literature is more described as a JAK inhibitor rather than a TYK2 inhibitor. It does have a bit of a effect on TYK2, but you know, JAK and TYK2 work together. They're signal transducers, so they work together. So we probably inhibit both at the same time with baricitinib. But the idea was, you know, you have an interferon response between SARS-CoV-2. It binds to one of those receptors. JAK1, TYK2 is involved in there. If you could block it, you could decrease inflammation, improve mortality. In this case, oh, you can't see too well, I'm sorry. Yeah, there you go. Okay. In this case, in the GWAS, in one of the GWAS for COVID-19, you actually have a nice locus zoom. Locus zoom just, there are these plots which are actually just zooming in on the GWAS specific location. See here, it's up around TYK2, and you can see that around the TYK2 gene location, there's a nice signal for severe COVID-19, which is roughly the same as this signal for TYK2 expression, so RNA production. Okay, so that was interesting. And if you use MR of expression of TYK2, so the more you increase the expression of TYK2, the more people die, at least using MR. And so if you do the opposite, so if you block TYK2 in an RCT, you improve outcome. What's interesting about this one is, so this was the Nature paper with the GWAS that found first the TIC2 signal. And the guy at the end, Ken Bailey, was also, so he's a clinician scientist and intensivist in the UK, and he was also on the steering committee for the recovery trial. So what's interesting about this one is as soon as they found that MR result, he actually put it into the recovery trial. So for this one, we know that the recovery trial and the result of baricitinib saving lives was directly influenced by the Mendelian randomization and genetic studies. And then the last one, which never really caught on because it was at the end of the pandemic, is interferon lambda, which is also called interleukin 28. It's not a true interferon, it's not a true interleukin, whatever. And the idea here is also there's a big inflammation cascade. There's different cytokines being produced, but the end result of interferon lambda is what's in the red square there, MX1, which we're not going to talk about, but OAS1. So if you give interferon lambda, you increase the level of OAS1, sorry. And what did we predict? This was with Sirui and Tomoko and Tianwan, was he in this one? I forget, but anyway. This is what we found. So at the top, this is actually the more you have OAS1 circulating in your blood, the better the outcome. This is the expression of OAS1. The power is a bit limited, but you can still see something. And then the splicing, which we're not going to go into because I think Sirui might have a look at splicing, but we'll go. The point is the more OAS1 in your blood, the better the COVID-19 outcome, which is again supported by the same kind of locus zoom and co-localization in this case, if you're familiar with it. And this is exactly what they found. So in this New England trial, New England Journal of Medicine trial, they gave interferon lambda at the start of COVID-19. So someone presented with COVID-19. They directly gave them interferon lambda, boosted their OAS1 level, and it prevented hospitalization and death. All right, so quick recap. We have good negative controls, at least the ones that I published, because I'm sure there are more negative controls out there, but it's harder to find a negative in the literature than a positive. But there's good negative controls, that's for sure. So MR, if it's done well, most often you shouldn't predict something that's not going to be positive. That's all. Yeah. And then if you look at the three trials that I showed you, well, really MR and the trial are saying the same thing. Now the question is, I told you there were 6 classes of medication that we use that were proven to work by trials. There's 3 other ones, which is just I want to end with this limitation of MR. The 3 other ones are antiviral at the bottom, and corticosteroids and anticoagulants. The steroids are probably the first class of medication that was really shown to work. Anticoagulant, the evidence is a bit mixed, but it's still there in a lot of guidelines. But the point is, MR is really only good if you have if your genetic variant is a determinant of a very specific phenotype. And both anticoagulant and corticosteroids have such a large biological effect on they encompass so many genes and proteins that we just don't have good, valid genetic instruments, and most of them would bypass through that assumption at the bottom that it doesn't skip the gene or doesn't skip the exposure. It would go straight to the outcome, so that's what you don't want. So for those 2, well, it's not that it's not that we couldn't try to run an MR, it's just that it would be completely invalid. Okay. And for antivirals, so those are drugs that directly kill the virus. Well, our body doesn't produce them, so there's no genetic variant to determine them. Now, you could argue that you could maybe do a pharmacogenetic study, for example, but that would be a bit complex. These haven't been done, and you can think of many reasons why, but yeah, it's unlikely that it's going to be done on a very large scale, and it would be complicated to put in place. Because you need to give the drug and then watch the different level, but then at the point, if you're already giving the drug, why don't you just run a trial of the drug, so it's. Yeah, so for infectious diseases, or for any, well, it's really only infectious diseases. Where the disease is produced by something that's not from the human body, well, and the drug kills that, and it's also not from the human body, you can't really use MR, but those are the only three, so it's not like. Yes.
Still online, which is great.
But I don't think they heard me though.
No, because it crashed, when I came in, it crashed, so
Okay.
if we could,
Just turn the sound on here.
Yeah, yeah.
it's on.
at the top.
Yeah, and then if you just reshare your screen,
Oh,
because
yeah,
it seemed
yeah.
to drop the,
Yes.
sorry about that.
No, that's fine.
Okay, so someone online says that they missed about 20 seconds.
Yeah, that's about it.
Yeah.
From the time I went back and forth, so about 20-30 seconds.
You guys met the conclusion, which I was just saying that MR is great, you guys should do it and join our lab and have fun.
Well, thank you very much.
Thank you.
But now we'll open up for questions.
Can we go back a slide?
Go back a slide, yes.
So that's a pretty interesting statement, the lack of corticosteroids and anticoagulants.
Yes.
How is that threshold determined? Let's say that we have a drug that's right on the boundary between having a large biological effect and not.
Yeah.
Like, how do we know, like, yeah, we can use this for a month?
Yeah, there's no broad statement that can really answer this. For like, so for example, so for OAS1, what we did was look only at variants that was like very close to that gene, which are called cis protein quantitative track loci, so cis pQTL. And so with the assumption being that if the variant is close to that gene and it impacts the level of that protein, because it's so close to it, it probably doesn't, it probably only affects the level of that gene through like directly affecting the gene rather than being another chromosome and then another different pathway and blah, blah, blah. For corticosteroids, the problem is that there's so many receptors and so many proteins, and a lot of them are actually metabolites and not even protein. That's a steroids, right? They're cholesterol based. So, I mean, I wouldn't know where to start, and I don't think I've ever seen a good MR study or any MR study really with steroids. Vitamin D, which is not a protein, its biological pathway is super well known. So that's kind of the way we were able to look into it. So there's no all-encompassing way to do this, and keep in mind that MR is not always only used for like a protein or a specific drug target. Sometimes you want to ask like broader epidemiological question. And in those cases, it becomes even trickier. So if you do like an MR with body mass index, well, that encompasses a whole bunch of other genes and pathways, and then the arrow there, which is what we call pleiotropy, is even more of a problem. But that's why in one of the slides, maybe I can go back. Yeah, you see on the left, you can see, right? On the left, just try to, yeah. On the left, there's like multiple Mendelian randomization techniques. And those are used when you think there could be some pleiotropy. And so they're more robust to different assumptions. So the truth is that you're kind of stuck making different assumptions, and you're hoping through your understanding of the biology and the use of these different methods that they work. Yeah. Thank you very much. Thank you.
If there's questions from people who are online, feel free to put them into the chat.
Guillaume, I was wondering, what do you work on now? Because this is all password.
Yes, yes.
What's the research program moving towards now?
Yes, so, well, there's still a lot of work to be done on those PQTL and the cis-PQTL. But what, I've decided to, take a bit of a detour. So the main message there in this presentation is that you can do causal inference with genetics, okay? And so causal inference is stronger than just an observational association, and what we hope is that by using causal inference, you can translate this easier, so you can optimize the first two arrows to then make that third arrow more likely to succeed. So I've tried to develop different methods to causal inference at the HLA, which is a very nasty word, because it's very scary and very complex. But so that's what I'm mostly working on at this moment. But yeah, I mean, I'm still an infectious disease clinician, and so the UK Biobank is releasing soon, hopefully, it's some data on infectious disease, so I'm working on that too. So I'm still working on that with different diseases, and then do different methods of causal inference, but a different focus. So MR, for example, does not work at the HLA, like you need to do something completely different. So that's what I'm working on, amongst other things.
So tell me about vaccination in this context. Does vaccination change any of the conclusions? And how?
I'm sure it does, actually. So maybe you can go back. Okay, so I forget which one of these speaks at chromosome 6 it is, but one of them is the HLA. And what's interesting is they actually arrived quite late compared to other ones. And I'm convinced that part of it is actually because people became more and more vaccinated. And so you could see more and more genetically an effect of being vaccinated, and your response to vaccination will be partly determined by the HLA. There has been multiple paper actually, but the one I'm talking about was in Nature Med, where it was either DR or DQB1 that was associated with better vaccine response, and then if they had those observational cohort over time, and the same HLA allele that was associated with better response was also associated with less relapses. So we know for sure, and there's in the clinical ID field, there's actually a lot of questions right now about maybe redoing some of those trials now that people are vaccinated, and asking ourselves, does it change things now that you have HLA and antibodies working in your favor? Would we still see the chromosome 3 peak, which is, by the way, still a bit cryptic? We're not like, there's a lot of immune related genes in that focus, but we're not entirely sure how they all work together. So I actually think we would probably see less of an effect in many of these peaks, but it's never been proven. We're gonna have to wait 10 years and redo it.
Be interesting if they completely disappear and go to null. I don't know that they would likely do reverse direction. It would be interesting to see if they completely go to null.
Yeah, yeah.
And the other question is, have you seen changes with different strains of the COVID-19 virus, and are you even able to look at that?
Yeah, so I think we're not able to. But I mean, first of all, so that's what I was saying earlier, is like everyone has been kind of working in their own silos, which is good if you want to be honest and compare, but it's bad if you want to answer specifically those questions, which is what's the interplay between the host genetics and in this case, virus genetics? We don't know. But it would also be difficult, like, these kinds of questions have been answered in like, I think it was malaria, but malaria, so there's definitely an interplay between malaria genetics and human genetics. but malaria is a parasite, so the way it evolves is, the way it develops mutation is a lot closer to us, and their genome is like much closer to ours than viruses. Viruses, we like to go, so there's the Omicron variant, the Delta variant, it doesn't really happen like this. It's just a mess, and it just, eventually one of them becomes more, overwhelms the other epidemiologically, but, to just say, yeah, it would be so difficult to actually adjust with this. I wouldn't know where to start. So that's the other problem. But in other diseases, it's been done. I mean, malaria is the greatest example. There's definitely an interplay between the two.
For this one, I remember that it's 40 associated loss. I mean, is the green dots
Oh,
associated loss?
yeah, no, sorry, the green dots, the color of the dots doesn't actually matter. When I said 40, what meant the peaks. Okay, so, 1, 2, 3, 4, 5, and some of those peaks sometimes are very close to each other, so you can't really make the difference. Like at the chromosome 6, there's probably 3 independent signals there for sure.
Because it doesn't look like only 40 from the green dots and the
No,
some
no.
is below the threshold.
Yeah. Yeah, so we only counted above the red line. And yeah, the green dot, so one of the sanity check in the GWAS is if you see peaks like this, because the variants that are close to each other are usually inherited together. So if one of them is associated with the disease, then the one that's always its mate at conception will also be associated with it, right? And so that's what you see when you see like multiple points like this. Okay, that's all you see really. So that's why. So no, like if you're going to count every single point, there's probably a few thousands for sure.
And just a statistician question. Because in the theoretical statistics world, right, all kinds of people are developing fancy methods to do multi-locus MR, all resting on strong assumptions, which are completely impossible to
Exactly.
yeah, what do you think of them? Have you read some of those?
I know it's so there's when you look at like something that's very precise like a protein, I try to go back to the biology or at least the geography of the genome and keep it close. And I know it's cheating because this is like the whole point of doing GWAS to use to find your instrument is because it's hypothesis free and you don't like you don't have a target gene in mind and a pre-specified hypothesis. And I'm telling you, yeah, but you need to choose your biology and like look into the biology to choose. I know it's cheating,
Yeah.
okay? So it's circular logic, but sometimes it still makes sense. So I think vitamin D made sense. I think most proteins make sense. For bigger traits, but bigger traits in terms of like pleiotropy, BMI, I think this is the only time where those other like something like MR Presso and all those can make sense because you have so much. The assumption that the pleiotropy kind of cancels each other when you have 400 different variants makes more sense to me. But still, I try all of those techniques and publish all of them, and I try to be very conservative. And if it's only one of those MR methods that has a p-value that I like, I try to say this is a null result. I think that's the only thing that can.
What would your like number be that for something that's multiple locus? Like for example, if you had, sorry, I realize I'm like digging deeper into this, but like I just thought of it, like if you had something,
Yeah, exactly.
like 2 loci, right? Then like you can still use a lot of these traits, no, because you can say, okay, you can account for the second one, and you can do that, and then it's like obviously the more loci you get, the more complex it gets. But like, would you have a strong cutoff or like anything over 5? You not even think about, or
I don't think you can have a strong cutoff. Yeah, that's the problem with MR. Yeah, the main problem, the difficult part of MR is the pleiotropy. So, yeah, it's hard. I think it was another, and did I miss it?
Anybody online? I think last chance here. Oh,
No, sorry,
Benjamin,
yes.
yeah.
So I was just wondering, since COVID-19, have these approaches that you've discussed changed how we study other infectious diseases?
So unfortunately, the data on infectious disease is just starting to come out. So we haven't had a whole lot. I think there's been very few traits. Like hepatitis B, I've done something on. Hep C, I think there's been a bit of work on that. Tuberculosis, but they're all like limited sample size. And a lot of times it's the HLA that comes out and then everything falls apart in terms of MR. So, and I think the HLA comes a lot first because for a lot of those, we develop a strong immune response so it's just, but there have been work on influenza using the same kind of framework. And it's also, it was underpowered. We didn't have 40,000 cases. Like the big difference now is because it was a whole big consortium. So I think the main problem is still sample size for most of those diseases, unfortunately. But yeah, without spoiling too much, there's work done in the UK Biobank, because I don't know how much I'm allowed to say, but there's work done in the UK Biobank. We see other stuff, which is interesting, which is not all at DHLA, and hopefully we can do some MMR with this.
There's old studies in infectious disease where people were looking at families. So like everybody in the family had
That's it.
tuberculosis, everybody in the family got leprosy, whatever.
That's it.
Like, why did everybody get that?
So those are the inborn errors of immunity studies,
Yeah.
which, yeah, so that was what I was trying to say earlier. It's hard to say. So not to dismiss them, because there's valuable biological insight with rare variants, and one of the most successful new drug to hit the market in the last 20 years is PCSK9 inhibitor, which is, I think, the whole thing started by studying like a rare Mendelian phenotype. Okay, so I'm not dismissing this, but it's true that like if your inborn errors of immunity is you have absolutely no B cell and T cell function, clearly whatever we find in you cannot apply to people who have B cells and T cells, right? Like this is like completely different in my mind, completely different disease, and the treatment for one cannot be applied. Like treatment for a stem cell transplant, we're not going to do this for us, right? Like so.
Well, in that case, I think we'll say thank you very much, Guillaume.
Thank you.